Welcome to the second part of Update Annotations video. In this video we will show you how to create custom annotations using the Annotations Manager. Here I am using Rabbit as the non-standard organism for creating custom annotation in Strand NGS. I have downloaded the FASTA files and the GFF3 files from Ensembl for Rabbit. Before adding annotations for an organism, it is important to create a custom organism which is not present on the Strand server. To create a custom organism, click on the small arrow beside Create and select New Organism. Here you need to specify the details of the organism, that is common name, rabbit, scientific name, Ortolagus uniculus, taxonomy ID, 9986. If you have GenBank files in GBFF or GZ format from NCBI, select the checkbox, provide the build name and load the GBFF or GZ files. The build will be created automatically. If GenBank files are not available from NCBI, click on OK to add the organism. Once the organism is added, right-click on the organism name and select Add Build, and you will see three options using which you will be able to create the build. Choose the build options based on the files formats, which should be tab delimited, FASTA, or GenBank files. It is also possible to create the build, reference, and genes and transcripts annotations from one or more GenBank files, where each file contains complete genomic information for a chromosome. Since I have FASTA files, so I am selecting the third option. In this window you need to provide the name of the build, the source, in this case it was Ensembl, and you can also add any notes about the build in the description section. Browse the FASTA files and click on Finish. The build will be listed under the organism and HEG reference will also be updated under build. Similarly, to add annotations, right-click on the newly created build and select the option Add Genes and Transcripts. The supported file formats adding annotations are TXT, TSV, GTF, GFF3 and GBS. The annotation file that I had for Rabbit is in GFF3 format, so I am selecting the GFF3 option and clicking on Next. Here are relevant details about annotations, such as the name of annotation, source, any additional notes in the description section, and the version details can be added. Choose the file and click on Finish. This message comes up when there are certain entities that do not match the build. It will come first for the gene annotations and click on Yes. Then for transcript annotations, and we can click on Yes to proceed. The genes and transcripts annotations will be updated and listed under the build. Note that Go annotations will not be available in the gene annotations created from GenBank files. But one could export the gene annotations, populate the Go column based on common identifier, and then import back the updated annotations via Annotations Manager. Now we will be showing how to add a target file or say sure select kit for a custom organism. Select the build, right-click and say Add Targets. Here we select our target file containing the regions targeted by the sequencer or sequencing kit. The file formats supported here are BED, TXT, DSV and CSV. Now we can format our data. Say we need to remove some rows, just use the comment indicator and indicate the rows. It is done. We specify the header and say Next. This step here helps us to eliminate the rows by simply unchecking the corresponding box. But always remember we should have columns, chromosome, start, end, gene ID necessarily. Say Finish. As we can see, the pop-up says the target file has been imported under the selected build and also informs about the number of regions the list contains. Let's confirm. The annotations will be updated and listed under the build.